Should I start the screen sharing?
Hi, everyone. Good afternoon. We'll give it just another moment and let others join the webinar. We'll get started here in just a second. Hi, good afternoon, everyone. My name is Chris Boudreau. I'm Manomet's Director of Development. Thank you so much for joining us today for an exciting look at the technology that empowers our scientists. If you're new to Manomet, for over 50 years, we've been a leader in bird research and conservation. We use science and collaboration to strengthen flyways, coastal ecosystems, and working lands and seas across the Western Hemisphere. We do this work with many partners to help nature and local communities thrive. Today, we're joined by two very special guests, Dr. Shiloh Schulte, Manomet's Senior Shorebird Scientist, and Dr. Marissa McMahan, the Director of our Fisheries Division, who will share some of the ways that they're using technology to advance their work. Just a couple of quick housekeeping items first before we really get started. At the bottom of your Zoom screen, you should see a box marked Q&A. If you don't see it, use your mouse pointer to hover over the bottom and it should appear. Now, if at any point during the presentation you have a question, feel free to click on that Q&A box and enter it. We're going to pause after each of the presentations to answer as many questions as we can. And if you are unable to stay for the entirety of today's presentation, it is being recorded. We will send you a follow-up email with a link to the recording here within the next day or so. Finally, I'd like to extend a special thank you to Howland Capital and Hemingway and Barnes for supporting our online educational opportunities like today's webinar. And now, without further ado, I'd like to turn things over to Manomet's President, Lizzie Schuler, to get us started.
Hi, thank you everybody for joining. It's really a pleasure to be sharing this information with you. And as Chris mentioned, so we're a small to mid-sized science-based organization with over 40 people sprinkled throughout the Western Hemisphere. And we are, oh, I realized I need to, can I share my video? I don't know if you're seeing me or not, but I'm not seeing me. So I hope you're seeing me. But anyway, as you'll see, oh, here we go. Okay, there I am. As you'll see, certain technological innovations really are giving us exponential understanding of where and how and why birds and fish are using the habitat that they're using. So we're thrilled to share with you what we're doing and also really invite input and thoughts about how we can ramp up this work. We want to share with you what we're doing and what we know, but we are looking to learn more, and this is really an invitation to have you all help us learn faster, because in this day and age with habitat loss and climate change, we really need to learn as quickly as we possibly can. So thanks so much for joining, and Shiloh, over to you.
Thanks, Lizzie. I appreciate that intro. And I will go ahead and, it think we need to add my video, but we'll just go ahead and get started. So we work with, what I'm going to present to you today is our work with mapping global bird migration with GPS tracking. This is one aspect of of how technology intersects with our work to conserve shorebirds and learn more about their ecology, their migration, threats to different species, and how we can conserve this species group for the long term. So just a little bit about shorebirds before we get going to orient everyone who may or may not be a shorebird expert. We're specifically talking about the group of plovers and sandpipers, not so much the other birds that use the shore like gulls and herons and quite a range of others, but specifically this group of shorebirds, and in particular those that are long distance migrants. Shorebirds in particular excel at migration over huge distances. This map in the center of the screen shows the migration pathway for one shorebird, the American golden plover, and golden plovers nest in the Canadian and the Alaskan Arctic, and then migrate nonstop from either eastern Canada or northeastern United States over the Atlantic Ocean to South America, and that can flights of up to 5 or 6 days nonstop to landing on the northern shore of South America, and then proceeding on to Argentina and the grasslands of central South America. And then in the spring, they do it again, but as you can see from the pathway on the screen, they move in an elliptical pattern. So coming back up, they don't cross back over the Atlantic, but rather up through Central America and then through the mid-continent back up to their breeding grounds. And a lot of this has to do with wind patterns at different times of the year, but also food availability. And so this incredible distance traveled is an advantage for the bird because they can take advantage of the abundant food resources and few predators in the Arctic in the summer, and then shift and avoid the winter months by heading to South America. And really, they can do this over a relatively short time. Think of flying for 5 days nonstop is impressive, but it also is relatively short over the period of a year. So they can quickly move from one end of the hemisphere to the other, and then take advantage of different food resources. But this puts them at significant risk as well. So many species of shorebird, including golden plover, are in decline because these long distance migrations that are advantageous are also very risky. So if these birds rely on just a handful of habitats throughout the course of the year and specific locations throughout their migratory pathway. And so if they lose one of these sites, that can be disastrous for that individual, and then cumulatively for the species, and then across a large landscape for multiple species. So things like oil spills, development of the coastline. You look at this image and you can think that much of or all of the Atlantic coastline and used to look like the island in the bottom of the picture, and much of it now looks like the island in the top. And so if a shorebird's coming in expecting to see the island they may have landed on for the last 10 years, now it's covered in houses or has some other development going on, that's a huge change. And there's other things like pollution, change in predator communities. Some places the shorebirds are hunted, and then there's constant disturbance. So even where there are places where the islands are not developed, they may be used recreationally, and these are all threats that these birds are facing throughout the year. And that's during migration, and there are additional challenges at the breeding and wintering sites. So our mission here, we're going to talk about today, is how we identify what those migration routes are, the specific stopover locations, and then some even more fine-grained detail at like how the birds are using those stopover sites while they're there, where they roost, where they rest, where they look for food, what do they do in response to predators. There's a lot of things we can learn through the use of technology, intersecting with what our desire to understand these birds and conserve them, protect them for future generations and for themselves. So one of the tool that I'm going to be talking about today primarily is the use of GPS trackers. So shorebird migration is very difficult to track. Shorebirds nest often in very remote locations. They're very, very fast. They can fly. We can't. Very hard to catch them. So you have to, in order to find something out about them, you have to put your hands on them in order to put a tracker on or a band. And for many years, and still currently there are banding birds was our primary source of understanding how migration works. And we've learned a huge amount from putting little color or metal leg bands on birds and then either recapturing them or resighting them at other locations. And that's a very useful tool, but it has a lot of limitations. You have to be where the bird is in order to see it. You have to get that report back. And if no one's looking at a location, you don't learn anything about it. And again, shorebirds are often at these remote sites. They also change their weight a lot. So prior to migration, a shorebird can double their weight, and then they lose that. They go back to their pre-migration weight or lower by the time they finish these epic Atlantic crossings. So in the course of 5 days, they may lose half their body weight. And then they have to put it all on again and plus some in order to make their next section of their journey. So the fact that their body weight size changes a lot during the migration is a challenge to attaching some sort of tracking device. However, there's been relatively recent developments in GPS tracker and battery technology that have opened up a new range of possibilities for our work. So back in 2009, the work really kicked off with larger shorebirds with the development of 9-gram tags. And that was a result of the miniaturization of the components of the tags, but also the more efficient batteries that could last a longer time in a smaller scale. And this change allowed us to put trackers on these larger shorebirds that were moving long distances. And that work was initially pioneered by my colleague and coworker, Brad Wynn, back when he was working in Georgia. But now it's much more widely used. And as currently the GPS transmitters are down to as small as 2 grams for some more limited applications. And those are being deployed on some shorebirds. They have some limits, but the real advantages are the smaller size of the tags now, but also this connection with the Argos satellite network that allows us to track them multiple times per day. And so we can really, and get the feedback on a daily basis. So we can really see where these birds are moving in almost real time. And so we can learn where the hazards are, where they're moving, and then we don't have to actually recapture the bird in order to see where it went, or even to see it again at all. So for Manomet's research with using these tags, we primarily use these 5-gram solar tags, and so you can see in the image on the side they have these little solar panels which actually recharge the battery each day. And this is why they have to be mounted like backpacks so they can see the sun. And we've assisted in the development of this harness method, which specifically for these shorebirds that we work with, that works a couple of loops like a backpack, but instead of going over the wings, it goes up and over the legs of the bird. And the bird's legs are, I want to say constructed, it's probably not the right word, but they're positioned differently than a human or biped or a mammalian biped, in that these leg loops can be attached securely to the bird without impairing the leg movement or the wing movement. And they sit on the lower back of the bird, which allows the bird to move freely, but also allows the battery pack to charge and transmit effectively to the satellites. So it's been a very effective system for tracking these birds, and it's fairly quick to attach once you know how to do it. So in addition to the tag, we also continue to put bands on the birds or little flags on the birds, so there's an independent way to assess what that bird is, because if something happens to the tag, falls off over time, then we can still identify that bird through the band. We take a number of other measurements to assess body health and size in comparison of birds in different parts of the range. So I'm going to talk today a little bit more about one species in particular that we're working with, and that's the whimbrel. And so this image right here is the Whimbrel's nesting habitat up in the Arctic. And I took this video at one of their nesting sites. You can see the eggs here in the foreground. And let's just watch for a second. This is your introduction. If you don't know anything about Whimbrels, here's your introduction to Whimbrels. And for reference, this bird is about the size of a crow. Maybe a little bigger. So this bird will sit on these eggs for 25, 26 days, I think, is about the normal incubation period. So she sits there hidden in the grass on this open tundra landscape. And then when the babies hatch, the parents take care of them, primarily the male. And our method of capturing, we actually capture them at the nest using a bow net trap, which is like a soft net. And that's when we capture, or at least one of the ways we can capture them and attach these GPS trackers. So the location here is marked with that blue star. It's up in, well, it's a little off here, but it's up in northern Alaska. There's been a range of projects working with Whimbrel in Alaska, but our work is actually over in the Arctic National Wildlife Refuge, which is this area in the far northeastern part of the state. And we're tracking them. I will show some more images of our tracking pathways from the birds we've captured. But it's a very important breeding area for Whimbrel. They nest there, they raise their young there, and then they head to where they spend the rest of the year down in the tropics, Central and South America, primarily. It's a very short breeding season. They arrive at the end of May, and they'll leave either in July or August, depending on how successful they were during the nesting season. And the uplands of the Arctic National Wildlife Refuge provide this nesting and staging habitat for Whimbrel. And the Whimbrel, as we'll see, travel throughout the hemisphere and return back to this breeding site. So it's very important for birds, both from the East Coast and from the West Coast. And then during the breeding season, they're eating a range of things, but in a large portion of their diet is from crowberries or other berries that grow in the tundra and these very low scrubby plants, and then a range of invertebrates that are using the tundra as well. So there's a lot of food up there for them. It's one of the major reasons they go there, along with the lower predator numbers. So they're just another example of maybe a better image of the nest and Whimbrel in flight. And this is a recently tagged Whimbrel over here that I'm about to release. I don't know how that happened. Video is inverted. Oh well. But yeah, we release these birds. They take off and off they go. And now this is the kind of data we can get back from our tracked birds. These are the tracks of two Whimbrel that crossed the length of the Pacific Ocean from Alaska down. One landed in extreme southern California, and the other, this bird in yellow, is named Dryas, goes all the way down, crosses over and into Mexico over the course of 3 days of nonstop flight. And if you see on this image, the green is wind patterns, and you can see these storms that they're having to navigate through and around as they migrate down the coast. They're finding these southerly wind currents, but you can see this one in yellow. This storm is forming off the coast of Mexico, and it makes a hard left turn and dives into the coast just ahead of that weather system. So they're very good at being able to find the right wind patterns and avoid the wrong ones. And they're not always successful at that, but certainly over time they presumably get better at it. And to watch this again in almost real time, as I was updating on an hourly basis to see when we get the next update from the bird, and suddenly you see the jump to the next location, and then overlaying the wind patterns, you can really see how they're working. They're working the weather to get where they want to go, and using that. In some cases, I think we had one that was going over. We had a couple times where these birds were flying at a ground speed of over 100 kilometers per hour, although their flight airspeed was probably about 55. Their standard speed during migration is about 55 kilometers per hour. but with such a strong tailwind over ground that was moving over 100 kilometers an hour for a short period because it found such a good tailwind. And so that obviously can be a huge difference during these long migrations. And then this is another image of tracking 6 of our birds from the Arctic Refuge, and each time step is 1 day. So over the course of a couple of months, these birds are making these huge journeys. You'll see that some of these birds are going down the west coast, on the Pacific coast, but there's a couple that are going over here along to the Atlantic coast. This one in black now crossing the Atlantic all the way down to Brazil. Its neighbors from the same nesting area are moving down the west coast. And then we have another one, this green line, where one of the males jumped from the coast of Virginia down to Suriname. And the rest, meanwhile, the other birds are moving slowly down the west coast of Central America. So you can see there's some very fascinating different migration strategies that are occurring. And we were just literally finding this out week by week this fall as we're watching these birds leave the Arctic and travel to their wintering areas. And not to say we didn't know something about the general pathways, but we can find this sort of fine scale detail about this really important nesting area of the Arctic on the Arctic Refuge. That's a breeding site for whimbrel from both east and west coasts. And some of these 2 males that went to the east, the green line and the black line, were paired with females that ended up going down the west coast. So literally birds from the same nest going to different sides of the continent. So that's not the only project we have going with GPS tracking and whimbrels. For many years now, I'm not sure, 4 or 5 years at least, we have a project that's been based on Cape Cod, and whimbrels use the outer arm of Cape Cod as a stopover site. I mentioned these really important sites that during migration that they need to be able to fuel up for the next stage of their journey. Cape Cod is one of those, and so these whimbrel are coming into the marshes in Wellfleet, for instance, fueling up for several weeks, and then taking off and heading directly from there out over the Atlantic Ocean. And their next stop might be somewhere in the Caribbean, or more likely the north coast of South America. And so between the months of July and October, whimbrels are coming through in waves through the Cape and using these salt marshes and the beaches and eating a lot of fiddler crabs. And they're using those long curved bills to reach into the fiddler crab holes and pull out these crabs. And so it's really important. It's a really important area for juvenile whimbrel. We don't see too many juveniles to the south in migration to the south of Cape Cod. They're mostly from Massachusetts north and then going directly out over the ocean. But the Cape is a really important site. And very few people had been working with juvenile whimbrels before this project. They're very difficult to capture on the breeding grounds. They're very difficult to figure out how to put an attachment on that doesn't impact the bird. Our team has been able to do that. Alan Neidl and Brad Wynn have been heading that up, and I've been assisting them with that project. But they were able to successfully capture juvenile whimbrel in migration, attach these trackers, and then start following them through the first years of their life, where they're doing something very different than the adults are doing. So that's an important piece that we really didn't have a good handle on, and really could only understand through the use of these GPS tracking devices. And here's just a couple of images from the work on the Outer Cape. It's obviously a rough place to have to work. If you're privileged enough to be able to get out and see this site, then in the Wellfleet marshes, I would strongly recommend it, especially in the fall when the birds are moving through. It's a beautiful area, and it's fascinating to see these birds and their behavior in the fall. But we can learn some things like this. This was one of the adults that was captured. He's one of the more famous birds in our history here. Is this bird Ahanu that was captured on Cape Cod, and this is a complete track throughout the annual cycle, going from Cape Cod down to the coast, north coast of South America, over to a wintering area in Brazil down here, and then in the spring moving back along the coast. Crossing the Caribbean, crossing the Gulf of Mexico, over to the coast of Texas. And as I mentioned with the golden plovers, they have this elliptical pattern where they come over and then move up the mid-continent during the spring migration. And that's what this bird did, and then headed up to the Hudson Bay Flats, to the northern Canada on the west side of Hudson Bay, where where it nested, and then headed back in the fall, post nesting, all the way down to the northeast. And you can see hits the coast and makes a hard right turn, and then down to back to Cape Cod, because it knows that's where the good fiddler crabs are. And so this is the kind of information, the kind of fine-grained detail about behavior, movement, and important stopover sites that you can gain from these GPS tracking devices. So in addition to knowing where they're going, we're looking at what threats are. So once we know where the birds are going, we can start working with our conservation partners to identify threats, to publish these results so people understand what's happening and put the information into the broader scientific community. And then to build this information into things like conservation plans, into international partnerships, to focus on these sites that are really important for a range of migratory shorebirds, and hopefully conserve them for the long term. So for our next steps, we're looking to scale up the work in Alaska. We tagged 6 birds last summer. We'd really like to tag another 20 or so over the next 2 years, to get a much better handle on both movement patterns, migration routes, and then variation in migration between years. And that's going to require significant funding. We're actually working on raising those funds now to purchase the transmitters, which are about $2,000 each, as well as the significant logistical challenges of getting to the Arctic and actually setting up camp and finding these birds. It's quite an undertaking, as you can imagine, to do that. So we're working on developing the funding and the infrastructure to do that. We have a new project that's going to be a collaborative work with University of Oklahoma and the Fish and Wildlife Service in Texas, which, if we capture a bunch of whimbrel there, putting on these transmitters that allow us to track the whimbrel even more, track these birds every half hour instead of just a couple of times a day. We can look at fine-scale movement during migration. And then we're looking at developing a project in Central America with American oyster catchers and other shorebird, that some of these birds are moving out of the United States into Central America for the winter, and we want to understand that connection and where the birds that are migrating to Central America are nesting in the United States during the summer. So that will be another project we use these trackers for. And then this, as I said, this tracking data can be immediately used to improve conservation impact and efficiency. So it can significantly help and focus our conservation efforts. We have a number of partners. It's not just Manomet doing this. The National Fish and Wildlife Foundation provides a lot of our funding for this work, particularly up in Alaska. The Georgia Department of Natural Resources has been a long-term partner and pioneer with this work, as well as the Center for Conservation Biology. We have partners in Brazil, South of Brazil. We have, of course, the Fish and Wildlife Service has been a long-term partner and still is with with the sites in Alaska. And we've worked closely with Environment and Climate Change Canada as well. So some of those are some of our primary partners in this work, but there are many, many others, particularly when we start talking about some of the other species that we work with. And I think I'm going to wrap it there. And I'll take some questions. And
thank you, Shiloh. Remember, folks, if you have a question, you can find the Q&A box down at the bottom of your Zoom screen. Just hover over if you don't see it, and just type your question in. We do have a couple of questions, Shiloh. Julie asks, are the whimbrels pretty calm during tagging? She noted that in one of the photos, it looked pretty calm.
Yes, that's a good question. They are very calm. You'd think with that long bill, they could use it like a weapon or something, but they don't. And they are, once you have the bird in the hand, they are very calm. The bill is actually not sharp at all. Most shorebirds have a lot of nerve endings in the end of a beak, and it's somewhat flexible. And they do that so they can find food, whether probing in the mud or the sand. So they don't use it to fight or to fight back against you or each other very much, because that's more of a delicate tool versus a weapon. And they don't want to struggle, because the instinct when a predator captures you, which they would see us as a predator, is to not struggle, because that makes them bite down. So they relax and they wait and they wait. If you relax, and open your hands up a little bit, they will be gone. They just take off. So that's what they're waiting for, is their opportunity to escape. And that's why they don't really struggle. So it really helps because we can put the tag on, we can band and get them back in the air very quickly, and because we're not so much struggling with them. The babies do struggle because they don't really have that finely honed instinct, but the adults are very calm in the hand.
Louise asks a question about some of the communities that are around the sites included in the Wizard group and how we include these communities, and can some of the residents be trained to apply the technology?
That's a great question. So applying the technology in terms of actually capturing the birds and putting the tags on definitely requires some specialized knowledge and experience for how to capture a bird safely, how to attach a tag safely, so it doesn't impact the bird over the course of the life of that bird. But that's not to say that you can't work with people locally to do that. We certainly do. We have people from Cape Cod to Brazil to Alaska that live nearby that work with us on finding the birds, in some cases on assisting with capture, and certainly with monitoring them over time. And involving communities in conservation is hugely important to success. If you just go somewhere and catch some birds and then leave and don't engage with the people that live around these important sites, then you won't be very successful, because you're not working with, if you don't work with people to make sure they're valuing these birds, these locations, these habitats, to see the value in the inherent value in these things, then it makes conservation either challenging or impossible. So that's really important. And in many cases, the people living there do value these birds or these habitats, and can tell us a lot about their use, about their history, about their conservation. So it's very important to form those partnerships.
Terrific. Thank you so much, Shiloh. And I think now we're going to jump over to Dr. Marissa McMahan, who's going to tell us a little bit more about the technology that she's using in fisheries research.
Great, thank you, Chris. I'm gonna start my video and share my screen. Here we go. Yes. Okay. Ooh. All right. Well, thank you so much, Shiloh. That was a fascinating presentation. I learned a lot, actually. So we're just going to transition a little bit and talk now about how Manomet is using what's called environmental DNA. So at its most fundamental core, environmental DNA is used to answer the questions, who's there and how many. So all organisms shed DNA. You can think of humans shedding skin cells or hair, which contains their DNA. Similarly, marine organisms can shed DNA, often in the form of scales or gametes or waste products, etc. And all of that material floats around in the water column. So by simply collecting water or sediment samples, we can actually detect DNA. That is the environmental DNA part, and learn more about species distribution and population abundance. So essentially answering the questions of who's there and how many. So there are two common approaches for answering the who's there question. First, which is the image there on the left. First is where we're looking for one specific species. So we call this single species PCR. So we know in this instance, we know the species that we're targeting. In the case of this image here, that would be salmon. And we can analyze a water sample and determine whether or not that species is there. So looking at presence or absence. This is actually the exact same technology that's used to detect COVID-19. So when you get your nose swab, the swab is analyzed analyzed essentially to detect the presence or absence of COVID. So very similar in terms of the technology there. The second way, which is the figure on the right here, that we can look at this who's there question is called meta barcoding. So this is used really for assessing biodiversity. So in this case, we go out, we take a water sample, and rather than just looking for one single species, we can look more generally at everything that's there. So we can't always identify down to species with this method, but at the very least, we can get a broad sense of which taxonomic groups are present. So then the second question, the how many, this is really a more sort of cutting edge development of this eDNA technology that actually allows us to detect abundance. So not only can we tell if a salmon was in a particular body of water, but we can also tell how many salmon are there by looking at the abundance of DNA in the water sample. There's typically some groundwork that has to be laid, excuse me, in order to do this type of work that it really typically involves actually measuring DNA abundance with known quantities of individuals. So picture sort of controlled tank experiments where you know how many salmon are in the tank. You take water samples to determine what the DNA abundance looks like. And then based off of that, you can go out into the field and actually use those correlations to tell something about wild abundance from water samples. So this use of eDNA is a bit more involved and can be a bit more time intensive, but regardless, this is really a game changer in the world of assessing marine populations in particular, which are notoriously difficult to assess because you often can't see them and they move. So this is really changing how we think about and in our ability to actually go out and assess these marine populations. So I do want to just pause and emphasize, again, sort of the benefits of this technology, because it is really a game changer. This technology doesn't threaten animals or habitat, right? So that's a really important part of this new sort of technique and sampling process. It's non-invasive, so compared to traditional DNA sampling, which requires you to actually take a tissue sample from whatever species you're studying, it's also much cheaper and more accessible than many traditional sampling methods. So for instance, if you look at this picture on the left, this is what's called electroshock sampling, which is quite a common technique for assessing aquatic species, where electrical currents are actually passed through the water to stun fish so that they float to the surface and can be measured. And in this instance, in the photo here, it's taking dozens of people to conduct this sampling. But excuse me, imagine instead being able to just send one or two people out to take a water sample and essentially be able to collect a lot of the same information. So that is what this eDNA technology provides, an easy, affordable, and accessible way to gather information. So Manomet is currently working with partners from the Maine eDNA project, which is a 5-year, $20 million project funded by the National Science Foundation. The Maine eDNA project is housed at the University of Maine, but is actually a collaboration of many higher education institutions, excuse me, and NGOs. We are partnering with eDNA researchers at the University of Maine, as well as the Bigelow Laboratory for Ocean Sciences, to conduct our eDNA work. So what UMaine and Bigelow are really looking for are partners that can apply this technology to real world problems and can connect this technology with community partners and stakeholders, which is really where Manomet comes in. So we are specifically looking to utilize eDNA to inform resource management and conservation, as well as restoration. So these are just a few of the ongoing projects that we're working on right now, looking at how to utilize eDNA technology. Currently, we're working to improve shellfish management and conservation with eDNA. We're also looking to better understand the impacts of river herring restoration to coastal food webs and identify and track the distribution of red knot prey, red knots being an endangered species of shorebird. So I'm going to spend just a little bit of time on each one of these projects to sort of walk through how we're utilizing this technology in each instance. So first up, shellfish management and conservation. So here, as part of our efforts to really build resilience through fisheries diversification, we've been working with shellfish harvesters and managers to understand how climate change is impacting shellfish populations. In particular, soft shell clams, which are a really important resource in New England, have been in steep decline in recent years due to climate-driven increases in predation. So, we're seeing that there's been sort of this ecological and also socioeconomic impact because it's an incredibly important fishery in New England. At the same time, we're actually seeing that hard shell clams or quahogs, which in the past haven't been as valuable of a resource, are rapidly increasing in abundance and economic importance in areas like Maine. So we're using eDNA to better understand quahog populations by tracking spawning events. So this figure here is actually showing a similar example of how you can use eDNA to track spawning events. So in this example, we're looking at salmon and salmon spawning in particular. So essentially you would be taking water samples over a certain period of time and looking at DNA abundance. And you can start to distinguish baseline levels of salmon DNA when the fish aren't spawning versus the really sharp peaks that you can see on this figure in the abundance of DNA when spawning is occurring. So we're trying to do the same sort of thing, looking at quahog spawning, because once we can determine when and where these spawning events are occurring, we can also, not only better understand reproduction, but inform management and conservation efforts to really maximize the potential for these populations to thrive. So next up, the second project that I'm going to talk about is river herring restoration. So river herring are an anadromous species, which means they live in salt water and spawn in fresh water, making upstream migrations every spring. They're also a very important forage fish, which is essential for restoring the health of coastal ecosystems. But they've been decimated over time by dams that block fish passage, over-harvesting, and pollution. So river herring restoration began in earnest in Maine about 20 years ago with the removal of the Edwards Dam in Augusta. And it was really a nationally precedent-setting case. And so we've seen more recent restoration projects have followed on both the Kennebec and Penobscot rivers and many smaller drainages as well. And a lot of these projects have resulted in the production of billions of juvenile river herring now entering coastal waters each fall, although this is still well below historic levels. But as rivers are restored, we want to be able to track the impact of juvenile river herring production on coastal food webs. For instance, restoring forage fish like river herring is one of the critical steps required to restoring haddock, cod, and other ground fish. So we're surveying juvenile river herring using eDNA. It's proving to be, a little bit less challenging than sort of the traditional methods. So the photo there in the middle is showing two juvenile river herring in the palm of someone's hand. So you can see that they are very fragile and easy to damage at this stage. So sort of traditional traps or nets that are used to try to sample these fish can be somewhat damaging, especially in flowing water. So we're aiming to use eDNA to study the production of juvenile river herring in restored watersheds as an alternative to traditional sampling methods that could be damaging to these fragile juveniles. And we hope that understanding production in relation to the characteristics of watersheds will actually help us figure out which river systems produce the most juveniles and why, which in turn will improve river herring management and help prioritize restoration efforts to focus on those areas that are most likely to provide abundant populations and support sustainable fisheries. And then the last project I'm going to talk about is looking at red knot prey identification. This project is an example of using that meta barcoding approach. So throughout the last few decades, there's been a shift in the distribution and abundance of red knots within the Manomet Pleasant Bay system, which is on the southeast end of Cape Cod. But the cause of this shift is poorly understood. So one hypothesis is that the northward displacement of knots out of Manomet and into Pleasant Bay is being driven by the shifting distribution of their invertebrate prey. So to test this hypothesis, traditional methods along with newer eDNA techniques are being used to characterize the benthic invertebrate community in the Manomet Pleasant Bay system in terms of looking at the distribution, abundance, and phenology of prey species. And then in addition, we're also looking at the actual diet of red knots through both stable isotope and metabarcoding analysis of fecal samples. So the stable isotopes actually allow us to look at the relative abundance of carbon and nitrogen in the diet, and that can inform what types of prey are most important to the diet. And then the metabarcoding will allow us to look even closer at the species of prey that are being consumed. And this approach will allow us to document phenological changes in prey abundance and diversity in the Manomet Pleasant Bay system in tandem with red knot prey consumption. So ultimately, we hope that this will allow us to understand what's happening in terms of these species shifts and and also inform how we manage and protect habitat for this endangered species. Excuse me, I have a bit of a tickle in my throat. Okay. And so I think this last project we're ending on is really a great example of a pilot study that's going to allow us to test and perfect this eDNA metabarcoding method for food web and diet analyses, and then expand it to other species and geographic areas. So just to wrap up and sort of look into the future, we have about 1 million different ideas for how to use this cutting edge technology going forward, and several new projects in development that I didn't even mention here today. It truly is a game changer in understanding the distribution and abundance of species and is incredibly cost effective. So we are also in a unique position to test how eDNA can be utilized to improve conservation and management, which has not necessarily been the focus of this technology thus far. So along those lines, we want to expand the pilot projects that we've been working on. We want to continue developing new projects, both internally, but also with partners throughout the Western Hemisphere. And then, one really exciting new development is this eDNA field reader, which is pictured here on the left. This is going to make eDNA technology even more accessible. So with this machine, we can actually run samples and get near real-time results. So again, very similar to a rapid COVID test, this machine allows us to collect a sample, run it in the machine, and in about 15 minutes, determine what DNA is present in the sample. So this is opposed to collecting samples and preserving them and then shipping them to a lab for processing, and then maybe 6 to 12 months later actually getting your results. So we're very excited about this machine and that we were able to receive some grant funding to purchase one. And we're hoping to use it to expand our research and monitoring, but also to make this technology more accessible to stakeholders and communities and even educators. So I think this is going to be a really exciting opportunity for us going forward. Okay. So yeah, with that, I just want to thank everyone for joining, and I'd be happy to answer questions. I also encourage folks to visit our website to learn more about this or to reach out to me directly as well. Thank you.
Thank you so much, Marissa. We do have a few questions. Just remember, if you have a question, you can enter it in the Q&A box down at the bottom of your Zoom screen. The first question asked about how long the DNA is detectable within the water.
So it depends on many different things, actually. It depends on the species and the decay rate of the DNA that's specific to each individual species. It depends on things like how much exposure to sun or other environmental conditions there are. It depends on things like flow rate and currents. So oftentimes what we find is that we really need to know something about the system that we're working in and that we're collecting samples in to really understand, how quickly the DNA might be dispersed in the system or how quickly it degrades. And so oftentimes we'll do sort of, like a pilot study to just really understand that for each unique system.
And let's see, one other question. would you be able to use eDNA to detect the presence of a duck, for instance, if it was using that particular body of water, like a Barrow's goldeneye?
Yes, you should absolutely be able to. So one of the studies that recently was really fascinating to see some results on was done in the Jones River watershed in Plymouth, Massachusetts, where Manomet's headquarters is. And they actually they used this meta-barcoding technique, and they took samples in multiple different areas in the Jones River watershed. And they were actually able to detect, it was like over 100 species, and it was anything ranging from fish and invertebrates to, marine invertebrates to mammals, reptiles, amphibians. It was unbelievable, the results that they were able to get. So anything that had been utilizing that water, whether it was something that was a terrestrial animal that just came and took a drink of the water, or, anything that was in that area was actually detectable. It was mind-boggling. So yes, you should be able to detect ducks. Absolutely.
That's pretty incredible. How much is the eDNA technology? Is it expensive, like the field device? Is that something that's really expensive, and who's developing it?
So there's two companies that develop the field readers. The field readers themselves are anywhere between $6,000 and $7,000. So they're a little bit on the more expensive end. If for instance, for one of our projects, we're partnering with a lab, Bigelow Laboratory. And so we'll go out and take samples, which cost next to nothing, because we just have to have, a sterile container to put water in. And then we bring it to Bigelow and they process it. And that's very, very affordable. It's, something like maybe $20 a sample or something like that. The field reader is a little bit more expensive, it's more that one-time purchase, but moving forward, being able to actually process samples in that field reader, again, should be very, very affordable.
Let's see, I've got several questions coming in. One is, how does Manomet keep tabs on the various technologies that are out there? For instance, technology that's being developed by other organizations that we might repurpose and use for our own programs.
Yeah, Shiloh might also have something to add here. I would just say that a lot of it for the work that we're doing in fisheries and the technologies that we're, aware of or utilizing, a lot of it is through the partnerships that we have, and, being exposed to these sorts of things through our partners, or through, attending seminars or webinars or conferences and learning about these new techniques that people are utilizing. So that's hugely important, just the communication among sort of partners and networks.
Can human DNA be detected with this technique? And if it can, would that be an opportunity to understand human interaction and usage within the natural system?
Yeah, absolutely. Human DNA can absolutely be detected. That's one of the reasons why we have to have, the gloves and make sure everything's sterile when we're taking the samples, so we don't actually contaminate the sample with our own DNA. So yeah, it definitely could be utilized to understand sort of how humans interact with different environments. The caveat being that, if you have sort of an estuary that's at the mouth of a river where, there's a lot of runoff from, septic systems or sewers or anything like that, any amount of like runoff that would be coming into a river, you're going to detect all of that DNA and not necessarily know where it's coming from. So that is, one sort of limit of the technology, and why we often try to pair it with other forms of sampling or, other ways of collecting knowledge if possible.
Marissa, do you keep track of all the species that you find, like in a giant Excel spreadsheet or database of some sort?
Yeah, we definitely keep track of all of that. It's also, so there's a couple of different things. So there's something called GenBank. And so for all of the work that we're doing, that's either developing, species specific, what we call primers, in order to sort of detect really specific species in a sample, that all gets cataloged on what's called GenBank, which is an open access genetic repository, essentially. And then, yes, when we collect samples for metabarcoding, we keep track of all of the species that we're finding. We also, what's really interesting about this is that if you take a sample, so for instance, with our Shellfish project, we take a sample and all we're really interested right now in is looking for quahogs. And so we're going to just specifically look for quahog DNA. But that sample that we take gets frozen in a -80 freezer and is good forever, essentially. So 5 years from now, if we go back and say, well, now instead we want to look for mussels or we want to look for something else entirely, we can actually just take the exact same sample and reanalyze it for a different species. So that's another thing that's really, really fascinating about this technology.
And we have time for one more question. Are you able to differentiate DNA when it comes from, say, a tributary that flows into the system versus when it's actually in the system itself from a species or that had been within the body of water?
That's challenging. And so that's something that I think, again, because this technology is so new, that's one of the things that is, kind of still being worked out. And it's very sort of specific to the system you're working in. So as an example, there's a study that's going on at the University of Maine right now where they're actually caging fish in a river system and then taking samples at multiple locations downstream and in multiple areas in the center of the river, on each bank of the river, to understand how the signal of DNA is dispersed from where a known quantity of fish are caged. And so, studies like that are telling us a huge amount about how these signals look in areas where water is rapidly moving. And then, we can build off of that to start to understand, if we see a signal from, salmon, is it a signal that's coming from 10 miles upstream, or is it a signal that's from right there in that, bay or area where you're taking the sample from? So yeah, we're getting there. But again, it's sort of specific to each of these different sort of systems that you're working in.
That's really fascinating. Thank you so much, Marissa. And thank you, Shiloh, for your insight and expertise on this. This was really terrific. And for those of you who joined us that are Manomet supporters, thank you so much. We really do appreciate your support. This is the type of work that you make possible when you support Manomet. So thank you. We're very grateful. If you have not supported Manomet in the past and you'd like to do so, now is a terrific time. You can visit us online at manomet.org/donate and make your gift securely before the end of the year, or you can scan this nifty QR code. So again, thank you everyone for joining us. We did record today's webinar and we will share a link to it tomorrow. We'll send everyone an email with a link to the recording. So please feel free to share it with anyone you think might be interested. And thanks again, everyone, for joining us. Really appreciate it.